DNA Depot welcomes you to Extracting Fruit and Vegetable DNA, Kit DD116. The objectives of this experiment are, first, to learn how to extract DNA from fruits and vegetables, and second, to see what DNA looks like and how to spool chromosomal DNA from a buffered solution. This hands-on activity will provide an opportunity for the student to appreciate the physical nature of DNA and the process of DNA purification by spooling it on a stick. Check your kit for the following items: extraction buffer, calibrated transfer pipettes, microtip transfer pipettes, wooden sticks, small test tubes, and large test tubes. In addition to these items, you will also need various fruits and vegetables, such as strawberries, tomatoes, and onions. 70 or 91% isopropyl alcohol, also called isopropanol or rubbing alcohol. Pencil with eraser. A knife. Place the alcohol in the freezer for at least 2 hours before starting the experiment. If you have a clean, small plastic bottle, you can pour some of the alcohol from the original container into the bottle and use this for all of your experiments. Don't forget to label the bottle with the chemical name and date. Place the fruit or vegetable on a cutting board or a pile of newspaper with a paper towel on top and cut a small piece of the fruit or vegetable. It should be the size of a pencil eraser. Place the piece of fruit or vegetable into one of the large test tubes. Use a wooden stick to push it into the bottom of the test tube. Use a calibrated transfer pipette and add 2 milliliters of extraction buffer to the test tube. To help release DNA from the cells, you will crush the piece of fruit or vegetable with 2 wooden sticks until it is like pulp. Keep one stick for a later step. Let the extraction buffer lyse, break up, the cells for at least 2 minutes. With a calibrated transfer pipette, push any large pieces of pulp to the bottom of the tube. Remove about 1 milliliter of the cell extraction solution. Be careful not to take pieces of the fruit or vegetable and transfer it to a small test tube. Open the bottle of freezer-cold isopropanol. Use a microtip transfer pipette to transfer about 1 milliliter of freezer-cold alcohol to the cell extract. Hold the tube at a 45-degree angle and add the alcohol to the cell extract by letting the alcohol slowly run down the inside wall of the test tube. Alcohol is less dense than water, so it will form an upper layer. The junction of the 2 layers is called the interphase. Step 9. Take a wooden spooling stick and submerge one end just below the interphase. Gently twirl the stick several times in a circular motion to spool, as if to wrap the DNA around the stick. DNA is not soluble in alcohol. Therefore, as you introduce the soluble DNA to the alcohol, it will start to form a translucent, gelatinous precipitate. Continue to twirl until you get a viscous, clotted mass around the stick. You can let the DNA dry on the stick and redissolve it in a smaller volume of buffer for further studies, such as separation on gel electrophoresis. DNA is present in all cells, and it directs all cell functions. You have just performed a DNA extraction experiment. The basic steps are: 1. To physically break up the cells. 2. Remove the unbroken cells and cell debris. 3. Add freezer-cold alcohol to precipitate or bring the DNA out of the solution. And 4. Extract DNA out of the solution by spooling it with a stick.